Hello everyone, and welcome back to this Nanophotonics and Plasmonics course. Today is the second and final video for chapter 3, and this will focus on confocal microscopy and near-field microscopy. So before getting started with those optical microscopies, let's focus on the concept of spatial resolution and localization. So localization is the precision with which you can determine the position of an object. This is not to be confused with spatial resolution, which is the ability to distinguish two separate objects from one another. So, although they're oftenly interchanged or confused, they are still connected. So, before the birth of near-field optics, it was believed that the diffraction limit was actually imposing an hard boundary on the spatial resolution, which could not be better than lambda over 2. However, we need to understand where this diffraction limit comes from in order to understand what we can do to beat this diffraction limit. So we have to introduce the concept of point spread function, which is simply a measure of the resolving power of any optical system. Typically, the narrower the point spread function, the better the resolution. So this point spread function is basically giving you the spread of a point source. So if you have a point source, say you have an electric dipole, which is placed on one end of this a given optical system, which is composed of optical lenses and everything, and you want to determine what's going to be the image on the detector, which is based in the image space. So this point source, represented by a direct function in space, if you take the Fourier transform and go in a reciprocal space, you end up therefore with an infinite spectrum. However, the evanescent waves that are going to be formed at the point source will vanish rapidly because they do not propagate, and those evanescent fields are lost before the detector. On the other end, when you have large K vectors that are basically not collected by the numerical aperture of the detector, you're also losing those K vectors. So in the end, the detector is actually collecting only a finite spectral bandwidth. You inverse Fourier transform, then you do not obtain the direct function that you had for the point source, but therefore you obtain something which is going to be spread in space. And that's the point spread function. So the point spread function is no more than just the spread image of a point source. So you can actually calculate this if you use dyadic green function, because you have a point source. You want to calculate the field distribution, the field intensity, E square in the image space, and you can use a parallax approximation to limit to only small angles. And then this is what you obtain. We're not discussing extensively this. I just want to highlight that this is dependent on the numerical aperture of the system, of the magnification of the system. You have the wavelength used for the optical excitation, and the media, the refractive indices in the object and image spaces. So if you have a point source which is a dipole oriented along the direction orthogonal to the optical axis in the image plane on the detector, then you obtain something which is broadened. So you have this Gaussian-like shape, which is given by this expression above here. And then if you have the dipole oriented along the optical axis, then you obtain this characteristic two-lobed profile. So this is something that also allows to determine the orientation of your electric dipole of your point source. You can do that experimentally. You can determine the width of this point spread function, which is also known as every disk radius, similar to the diffraction limit introduced by Abe, where you have now this magnification. You can also calculate or measure the depth of field, which is just the point spread function length or axial width, and that's going to give you how spread is your image along the optical axis. If we have two objects that are placed close to each other in space, end up with two overlapping point spread functions. So it's going to be difficult to distinguish the two objects. So how can we actually distinguish them? So you can look at the theoretical limit if you consider an infinite spectrum of k-vectors. And this is what you can get for the minimum distance if you want to resolve them. Diffraction limit, something very similar if you consider now the numerical aperture. And if you consider the Abe's limit, which is just we would discuss, the paraxial point spread function framework with two electric dipoles that are oriented along the transverse direction, then you obtain this expression that we already introduced. So it's basically lambda over 2. So how can we actually achieve better resolution than this? Well, simply just using evanescent components. As I said, those evanescent fields are lost before the detector, so if we are able to collect them, then we should be able to increase the spatial resolution and decrease the point spread function width. So this is the whole purpose of near-field optical microscopy that we'll discuss in a moment. But before going into near-field microscopy, it is important to discuss far-field optical microscopy, and in particular confocal microscopy. So confocal microscopy, still a far-field technique, allows to still achieve better resolution than the diffraction limit. So this is a technique which is widely used in sciences, from solid-state physics to biology to chemistry. And the principle that has been introduced by Marvin Minsky is just relying on the fact that if you illuminate a sample with a focused light, which originates from a point source, and you direct the response from the sample to this excitation onto a pinhole, then you can achieve a resolution that goes beyond the diffraction limit. So this is a picture of the first prototype that Minsky developed in 1955, and this is the first page of the patent that he filed in 1957, showing all the optical setup. Nowadays, this is what it looks like typically. So it looks like a regular microscope where you place the sample here, and you have optical excitation, and then you can have a CCD camera for the detector, and so on and so forth. So how does it work? We have highly focused beam. So we have the focus beam from a laser onto this pinhole that's going to give you the point source I was just mentioning. This point source will actually send light onto a sample through a microscope objective, which collect also the signal, the response from the sample, and this is focused on this pinhole before going to the detector. So this is exactly what we have. We have this sample being illuminated with a focus beam coming from this point source, and then not all the signal will go through the pinhole to the detector. Some will be blocked, but the signal that will actually go through the pinhole and being detected will give you a very narrow, or actually narrower point spread function than the non-confocal counterpart. So this has been derived in a variety of spectroscopy techniques, from just scattering to fluorescence to third harmonic generation, and even Raman scattering. So this is something very general that can be applied to any type of optical spectroscopy technique. You can actually see the whole mathematical formalism in textbook section 4.3. I will just highlight the major steps. So if you have let's say you have a molecule which is placed in somewhere in the object space. You have this point source which is focused nearby or onto the molecule. This gives you an excitation electric field in this object space. So if you have this excitation point spread function in the object space, then it's going to interact with this dipolar particle or molecule via the polarizability of the molecule. This will induce an electric dipole, which will in turn emit radiations. You can calculate what's the electric field being generated by electric dipole via the green tensor formalism. This field distribution that you get in the image space is the detection point spread function. You can also extract from there very easily the actual confocal signal, which is just simply the intensity that you detect by taking the square of this electric field. So if you use a Gaussian beam in a fundamental mode to excite the particular molecule, you can actually show that in the confocal signal, you have actually a higher intensity. So this is an integral that describes the excitation point spread function, the polarizability. So you see that you have this additional square that show up as opposed to a non-confocal signal. And this is the resulting from the existence of the pinholes in this confocal microscopy setup. And that is basically resulting in an increase in both transverse along the transverse direction and longitudinal along the optical axis resolution. We have a comparison between just conventional non-confocal microscopy and the bottom confocal microscope images of the same systems. You see that you have a clear improvement on the spatial resolution for different objects, such as a sunflower pollen seed fiber here, or human cells here. So let's go back to this concept of localization and position accuracy. So if you have a dipole emitter, like a fluorescent molecule, you can obtain a characteristic point spread function in the image space, and vice versa. If you know the point spread function, you can actually go back and reconstruct the emitter and determine its position. So typically a fluorophore can be localized with a precision of a few nanometers. And how is this possible? Well, you can use different techniques to actually be able to distinguish between the photons that are arriving at the detector. So you can select the photons based on their energy, polarization state, or arrival time, and you can actually separate the images even though they're overlapping. So this is a simple example. If you have unfiltered image from the detector, if you see a large blob like this, you cannot know that you have actually two molecules that are in this small region that are being imaged. But if you filter the polarization of the light emanating from those two molecules with two polarization states, you can actually separate one from the other. So these are basically just two molecules that are oriented in different directions in space, and therefore they will emit light with different polarizations. So you can actually filter out the polarization signal coming from each of these two molecules. So this is also why localization and resolution are actually not the same thing. So here we have a high localization power on this setup, although the resolution is actually not. So now diving into near field optical microscopy. So first, far field spectroscopy, how does it work? You basically send a far field signal and you focus the signal using a lens onto the sample, and then you collect the signal from the sample back to the detector. So as I mentioned earlier, so you're losing large K vectors that are not collected, mostly because you're losing all the evanescent waves. So that's resulting in a resolution limit. Now, in a near field, you basically get rid of this optical lens and you replace this by a small aperture. So you send this excitation beam on this metallic screen with a small aperture, and then you're forming evanescent waves at the surface of this metallic screen around the aperture, and then you use those evanescent waves to excite and interact with the sample. So in fact, you're retaining those evanescent waves, and that's going to lead to an increase in spatial resolution. So in principle, there are no limit to this technique. So however, there are challenges. So the evanescent waves do not propagate. It's just impossible to guide them toward the sample because just bond on the surface of this metallic surface. So in the end, you have to bring this aperture and the metallic surface as close as possible to the sample in order to have the maximum spatial overlap between those evanescent wave and the sample. But this in turn results in a strong coupling between this material that carries the evanescent wave and the sample. So the other thing which is also important is that the more you confine the fields, then the more rapidly they diverge. So if you try to focus the fields in space, so this is the electric field strength distribution, and you try to get it to smaller and smaller region, a smaller laser spot, then in turn, of course, you're going to increase the spectrum, the spectral broadening, and increase more K vectors, which is good. But on the other end, you're actually having fields that are decaying much more rapidly away from the metallic surface than if the fields were actually not focused. So because the fields are decaying more rapidly from the metallic surface, you need to bring them closer to the sample, and therefore you have an even more strongly coupled system. So let's look at light-matter interaction processes in the framework of a tip sample system. So this is something very general. So if you have a tip which is composed of a certain medium with the electric susceptibility chi, and you bring this tip in close proximity of a sample. So here you have a set of scatters which are composed of a certain electric susceptibility eta, and then you send this optical excitation. So this is an incident electric field that will interact with both the tip and the sample, and then you're gonna collect some scattering, and you have the total field that would be the superposition between the incident and scatter field. So in this system, if you have strong light-matter interaction, then you have to account for those effects. So you can actually look at an homogeneous almost equation for scatter fields, and this can be solved using the dyadic green function formalism. And this is the expression of the scatter field in this formalism, where you have the total field that interacts with sample and the tip, forming electric dipoles, and you have the green tensor, and that will give you the scatter field from this system. So if you can introduce some notations to simplify a little bit. I will not expand too much on that. We just have these SE terms that are just the integrals over the volume of the scattering part from the sample, and this T node E, which is just the integral over the volume of the scattering part from the tip. Okay, so you have the S and T functions even here. So this SE and T node E are actually integrals of tensor products. Okay, so but this is to simplify the notations. So the total scatter field, which is just given above, can be just rewritten in this way. So this expression is the exact same as that one here, just without the integrals, simplifying a little bit. So more generally, if you re-express the total field as a summation of scatter and incident field, you can express the total scatter field as a function of the incident field that interacts with the sample and the tip, and you have this infinite summation that comes into play. This is known as the Born series of multiple scattering process. So we'll see in the next slide what the physics behind that means. So this Born series that we just described here is in fact valid only for the tip sample system. So if you have a tip and a sample, now if you remove the sample and you have only the tip in the presence of this planar interface and the incident field, then you don't have this term coming from the scattering from the sample, and then you have this summation over the T nodes that show up, and that's the total tip scattering background. So if you assume that the scattering from the sample is fairly weak as compared to the tip, which is generally the case, you're looking at very small objects, you can really consider the scattering from this is fairly weak. You can actually expand this Born series into this smaller expression. And this is actually very important because this gives you the physics about the system. So this sum of T and S terms describe the different scattering events from the tip and from the sample. So we have a multiple scattering event that are just occurring in the system, and we're looking at the contribution of all those scattering events to the total scattering signal you're collecting. So the first term and the second term are just the incident field coming in onto the sample for the S and being scattered, or coming onto the tip and being scattered. So this is only one scattering event. That's what is shown in this figure A here. Now the third and the fourth terms, TS and ST, are two scattering events occurring successively on the tip and then on the sample, or first on the sample and then on the tip. So this is something which is pictured here on panel B and C. So panel B, the incident light scatters off the tip first, and then the scatter field from the tip scatters off the sample after. So you have two scattering events occurring successively here. Panel C is the other way around, so it scatters off the sample first and then the tip after. And then you can look at other terms. So the last term that is shown here, TST, is tip, sample, tip. That's why we choose those notations. It's easier to actually see those events. So the incident field scatters off the tip, back to the sample, scatters from the sample back to the tip, and then scatters being detected. And of course, you can clearly see that this is, of course, an infinite series, an infinite sum of terms. You can scatter back and forth infinitely between the tip and the sample. However, it is fairly obvious that the higher order terms become weaker and weaker and more negligible. So we can limit ourselves to the first few orders to get realistic pictures of the scattering signal from this tip samples system. So these are a few examples of such near-field probes used for localization of light. These are different families. So you have typically 3 families. The first one is transparent probes that are just dielectrics. Optical fibers are such a probe, so it's just made of glass. Then you have this aperture probe. You have a dielectric core, which could be just glass, an optical fiber, and then it's coated with a metal. In this particular case, it's aluminum. It could be silver. It could be gold. So the light is guided through the optical core, and when reaching the aperture here, then you generate evanescent waves at the surface of the metal. And then the last one is what we call an antenna probe, which are just nanoscale antennas that are placed at the end of either an aperture probe or a transparent probe, or even just a metallic probe. So this is a gold nanoparticle at the tip of an optical fiber, and this is a gold nanorod which is placed on this gold aperture probe. And these are just some images that can be obtained in this near-field scanning optical microscopy technique. So these are just a few examples showing that if you're fairly far away, 300 nanometers from your sample, you can actually have very low resolution. But as you bring your tip in close proximity to the sample, then you have more near-field interaction, and then you obtain much better spatial resolutions. And there's a comparison between topographic AFM images with near-field optical microscopy images for different systems, nanoparticle arrays or random nanoparticles that are 50 nanometers in size. So you have to remember that 50 nanometer gold nanoparticles is way below the diffraction limit of lambda over 2. So even if you use optical excitations with a wavelength of 400 nanometers, which is borderline visible range, then you can achieve spatial resolution with a diffraction limit in conventional microscopy of the order of 200 nanometers. So you see that you will not be able to resolve those 50 nanometers nanoparticles with conventional optical microscopy. So that concludes this chapter. We've seen a lot of new concepts with parallax approximations, focusing on fields, interface effects. We discussed about this spatial resolution and localization, introducing the point spec function, and then we discussed about confocal microscopy and near field microscopy, discussing this interaction series or Born approximation.